trying to understand how bacteria adapts across different environments. This is essential for combating infection, especially when we think about microbes becoming resistant to previous treatments. My field is comparative genomics and microbial evolution. I extract DNA from microbes that have been growing for long periods of time in different environments. There's so much about bacteria that we don't know, mostly because we haven't surveyed them in the right environments. The more times we put them in a new environment and start seeing different genes turn on, the more we understand what those genes do and how these bacteria thrive or perish in a variety of situations. The DNA extraction combined with genomic sequencing generates enormous amounts of data. So we use Mason, a supercomputer at Indiana University, which is managed by IU's Research Technologies group to process this mountain of information. In a fraction of the time, Mason can data crunch what would take the average computer days or weeks to do. Every time we publish our research, we have to promise to keep all our data. That way, other researchers can have access to our findings. They can then replicate our results or use the data to further their own research. All of our data is stored and accessible via the Indiana University's Scholarly Data Archive. Understanding microbes like E. coli on a higher level will help us work smarter when it comes to treating these structured organisms, lessening the probability that they'll become resistant to therapeutic treatments in the future. With the support of IU's Pervasive Technology Institute and UITS Research Technologies System and Tools, my research is more efficient and accessible. I'm Megan Berenger, and I'm a postdoctoral researcher at Indiana University's Department of Biology.